It's my honor to introduce our next speaker, Bluma Lesch from Yale University Faculty of Medicine, where she's an associate professor and she has her lab working on where she actually working on various things. The talk which she will have today would be about protamine lacunae and the role in sperm chromatin regulation. And yeah, floor is yours, Bluma.
Okay.
Nice to have you in this Fragile Nucleosome seminar series.
Thank you. Let me make sure everyone seeing the not center view?
Yes, it looks good.
Okay. Okay, great. So thank you for the introduction and thanks for having me. I'm really excited to talk to this audience and about this project. So my lab works on chromatin in sperm. And Yeah. So there's a lot of reasons to be excited about chromatin in sperm. One of them is the fact that it's probably one of the most extreme chromatin states that you can have. So I'm showing here, let's see, get the pointer. Um, so I'm showing here, this is just a DAPI stain of the seminiferous epithelium, which is where the sperm are developing, in this case in the mouse. And there's precursor cells at the bottom, developing into mature sperm that are going to be released into a lumen. And hopefully you can see that there are some really extreme chromatin transitions that go on during this process, including meiotic recombination, double strand breaks, homolog pairing. And then, of course, as you get into the mature sperm, there's this really dramatic transition where most of the nucleosomes get removed from the genome and replaced with small basic proteins called protamines. And that results in this extreme condensation of the nucleus. This is actually a highly regulated process that goes through a variety of transitional steps, including using some of these histone variants, such as specific histone variants that we just heard about. And it results in this really highly compact sperm nucleus. And I really love this picture. This is a human sperm and human red blood cells. And red blood cells, when they develop, compact their nucleus down pretty much as far as it'll go and then eject it and get rid of all of their nuclear material. Sperm do the same thing and they get down to this kind of like physical limit almost, but then they keep it and they're going to have to reactivate it after fertilization, which is a whole other set of challenges. So we're really interested in how this happens and the sort of various implications that it has for biology. Um, so I just said that nucleosomes get replaced with protamines, and that tends to be a little misleading, actually. So it's not just sort of another type of nucleosome that the DNA gets wrapped around. I want to emphasize what you're actually looking at. So this is a model of a strand of DNA with protamines in it. And protamines are these very small basic proteins that just pack into the major groove and neutralize the negative charge. So it's really just a fiber. It's not kind of a beads on a string kind of structure like you're used to thinking about. And what that allows is for the DNA to wrap into these really big kind of slinky-like structures called toroids that you can actually visualize by microscopy. That they're about 10 to 50 kb on average. And you can actually, this is a really very like physically rapid and spontaneous process. This is a paper from a couple decades ago where they put DNA onto a bead and then this is purified DNA, they add purified protamines, and they can actually watch in real time the DNA condense back to the bead as it gets protaminated. And you can see here the toroids starting to form, one, two, and then three toroids that fully compact the DNA in a matter of seconds. So this is like a very like powerful process. Um, in vivo, this doesn't take seconds, this takes days. It takes 5 to 6 days. And it's sort of unclear. So it's very highly regulated. It clearly doesn't need all of that regulation because DNA and protamines will do that by themselves spontaneously. So there's a major question in kind of like, why does it take that long? What's going on during that time? And what implications does that have for biology? Um, so this happens in the testis. Then mature sperm are in this kind of this protaminated form with mostly packaged in toroids. But importantly, there are some nucleosomes that are retained, depending on the species. This is anywhere from kind of 1% to 15% of the original nucleosomes. And we'll come back to talking about what that means. And then after fertilization, this whole process gets reversed in a matter of hours. So when we start to think about like why have the protamines in the first place, you could imagine that they're either facilitating something about sperm function itself during this time. Either they're important for the nucleus to condense, which maybe is required for the sperm to like get to the egg as efficiently as possible, to swim as fast as it can. There's a lot of other developmental things going on in the sperm. They have to develop a tail, they have to develop an acrosome. So it could be that this is all just kind of developmentally, structurally related to the sperm structure. Um, there are a lot of reactive oxygen species between the sort of male and the female reproductive tracts, and we know that protamines are involved in protecting the genome from that oxidative damage. And then there's a possibility that something about the proteinated genome packaging actually facilitates the initial entry of maternal histones after fertilization, and that having it set up in this kind of contaminated structure makes that transition easier and more efficient and kicks off the zygotic program more easily. So these are all kind of like intuitive things that sperm chromatin is important for and that protamines might be doing, and they all kind of make sense. We have not that much evidence for these actually being kind of necessary, these being the reason that we have protamines in the first place. So this is a major question of why we have this type of packaging. The flip side is, why do we have those nucleosomes? So I mentioned before, there's a few nucleosomes that are retained. They're definitely there. We can see them by Western blot. We can see them by mass spec. We can see them by chip. This is just a Western. So if you look, there's mouse sperm, human sperm. These are the histone bands. They're definitely there in the sperm. And there's a couple models that have been proposed. So this has been kind of like debated back and forth for a long time. A couple of models have been proposed about why this might be, and they kind of fall into two general categories. One is this is just an accident, like protamination is not as efficient as it could be, and it leaves behind a few nucleosomes. It just doesn't do the job that it's supposed to do. And this implies that like if everything was perfect, we would be fully protaminated. The more protamines you have, the better your sperm is going to function. The other option that's been proposed is that those nucleosomes are there for a reason. They're in a specific place or a specific sort of type of genomic material, like repeat elements or something like that, that they're important either for sperm function, something like motility, or for those initial stages of embryogenesis. And that implies that something that there are specific aspects of this protamine transition that are regulated. So you need to have nucleosomes in the right place or in the right proportions or something like that. And then if you perturb that, then you have worse sperm. So that the specifics of the regulation actually make for better sperm. So they make slightly different predictions. We have been sort of unable to resolve these models for a long time, largely because our technology is not really well matched to this unusual chromatin state that the sperm have. And so what I want to talk about today was applying Fiber-seq. So this is a much more appropriate type of technology to this question that has finally come out in the last few years. To look at the nucleosome protamine landscape in sperm. So we can actually see where the nucleosomes are relative to the protamines and try and begin to infer what they're there for in the first place. And this has been just a really incredibly fun and incredibly well-balanced collaboration between a student in my lab, Rachel Hewer, Andrew Sturgakis at University of Washington, who developed Fiber-seq, and Tommy Tullius, who's the one who developed the HMM, the Fiber-seq HMM, and has been adapting that for our sperm data. Okay, so just to back up for a minute, we are really interested. We've been interested in sperm chromatin for a long time. Our typical tools for looking at chromatin state across the genome are based on generally Illumina sequencing. And the problem is that short read sequencing, or we're talking about reads anywhere from like 30 to 150 base pairs, is just really not well matched to the structure, to the protaminated chromatin structure. So when you're looking at nucleosomal DNA, you're talking about kind of nucleosomal regions of 150, 200 base pairs or so. When you make these libraries, you're generally generating fragments of, 500, 600, maybe a KB. And so you can kind of get your sequence across that whole fragment and really resolve where it's coming from in the genome. If you take a 20 kilobase protected region and chop it up into 600 base pair fragments and then sequence those, you're going to get kind of a mess of reads just across this whole region, not really be able to see much enrichment either by something like CHIP for protamines, or something like ATAC if you're looking for like accessible regions. There's also some artifacts involved because this is highly compact. That's thing like the transposase-based methods sometimes aren't able to access where they need to access in the first place. Despite that, because this is a really interesting problem, there's been a lot of really beautiful and in some cases heroic efforts to use our existing technology to understand what the chromatin landscape, especially the nucleosome landscape, is in sperm. So I'm showing just two of the earliest examples. MNase-seq in pooled sperm. These are both at the HOX-D cluster. So this is an MNase-seq. You can see that there's enrichment for nucleosomes. There's some enrichment across the cluster. This is in pooled sperm. If you look across individual donors, you still see some enrichment. It starts to get a lot noisier. And it's hard to tell exactly where that noise is coming from. And then for CHIP, for histone modifications, in this case, it's actually CHIP, as well as ChIP-seq. You also see enrichment, but you also see a fair amount of noise. And so trying to sort of understand how that falls, what that exactly tells us about the sperm landscape has been challenging. And just to kind of drive home sort of the other side of this point, the other assumption that we tend to make in short read sequencing, most short read sequencing approaches, is that we have a population of cells, and we assume that when we see an average signal, it's representative of most cells in the population. And so if we see a pileup of reads over here, we assume that this region is open in most cells, or has a nucleosome in most cells. And this lower pileup means that either there's a mixture of cells where some have a nucleosome and some don't, or it's just lower extent of nucleosome retention. But we tend to make an assumption that this kind of is representative of all the sperm in the population, or all the cells in the population. What something like Fiber-seq or a long-read sequencing approach allows us to do is now kind of resolve this almost at a single sperm level, especially because sperm are haploid. Each of our chromatin fibers is going to be from an individual sperm. And this would give us a very different picture if now this, something like this sperm fertilizing an egg with a nucleosome here is going to do something potentially very different than this sperm fertilizing an egg with a nucleosome here, even though as a whole, as a population, we're getting a similar signal to here. So we think that this is going to give us a lot more information about what's actually going on in the sperm. Just to give you an introduction to what I'm talking about, this is the Fiber-seq method in general. For those of you not familiar, basically you apply a non-specific M6A methyltransferase, any region of the genome that's protected by a protein will not get methylated. And so then when you digest the proteins away and you do either PacBio or nanopore sequencing, you can look at where the methylation is, and then you infer that the sites that don't have methylation were protected by something or other. And you can infer what that was based on the length and the features of the protected sequence. When we do this in sperm, it's essentially the same thing. The only difference is that we had to modify the bench protocol a little bit because the chromatin is so compact. But then we get many long blocks. And so Tommy also modified the HMM so that we're able to pick that up and not just throw them away as artifacts. This is what the data looks like. Again, here's our, theoretical chromatin fiber, or theoretical read, PacBio read, M6A sites indicating that these regions were accessible. And then the sites that were protected, we assigned to either protamines based on their length, nucleosomes based on their length, or what we're calling subnucleosomal particles, which could be, small clusters of protamines or transcription factors or something like that. And then in between, there are regions that are actually accessible. So the first thing we wanted to do is basically we're saying we have long protected regions in our reads. We want to be, and we're inferring that those are protamines. We wanted to do some sort of validation to make sure that that was actually what we thought it was. So we just in vitro mixed genomic DNA with purified protamines at different ratios. And we can see from a blot that the more protamines we have, the less methylation we have, which is a good sign. So the protamines are in fact protecting the DNA from methylation. And now if we sequence these different ratios of protamine to DNA, we can see how that falls out along each of the reads. And this is, I'm going to show this kind of plot a lot. So just to orient you to what it is showing, each line here is a 10 kb read, a single 10 kb Fiber-seq read somewhere in the genome. So these are not aligned to a specific part of the genome, stacked up on top of each other. Within each read, regions that are called as protamines are in orange, regions called as nucleosomes are in green, and regions called as subnucleosomal are in blue. So in this particular case where we have a low protamine to DNA ratio, we see a few reads that have protamines, most are not protaminated. And as we increase the amount of protamines, we see more and more reads that are covered by protamines and fewer that are not. We don't note, just as a reminder, we don't have nucleosomes in this tube. So these regions that are being called as nucleosomes in this case are artifacts, and we do see some of that. We're able to account for it generally in the analysis. The other thing to point out, just as a side note, which I can come back to because I think it's really cool, that generally we see a read is either mostly protaminated or mostly not protaminated. We don't see kind of like half and half reads as we step up the amount of protamine, which implies that there's probably some cooperativity in how this is happening, which is consistent with what was seen before in vitro. Okay, so yeah, so this is what I was just talking about. This is this 1999 paper where they actually, based on this bead assay, saw sort of dynamics that they felt implied there was cooperativity. So we're confirming that nicely. The other thing that they predicted in this paper was that based on the sort of jerkiness of the compaction, was that there were going to be discontinuities in the protamine coat. And really excitingly, like when we look at sperm, we see something really similar. So our sperm are largely covered by protamines, as we'd expect. That's pretty comforting. But hopefully you can see that the sperm look different from the high protamine content DNA in that it's a little gappier. A lot of regions here that are actually accessible or fully unprotected, not even covered by nucleosomes. So we're calling these gaps lacunae. So, basically spaces in between where we see an absence of protamines. And sometimes there's something else in that, like a nucleosome. Sometimes there's not. Generally, we can now quantify the lacunae as well as the protamine blocks themselves. And what I'm showing here is just kind of a profile of what we see overall. Some of our reads are fully covered, fully protected, and those don't tell us very much. That's over here, as you would expect. If we look at the length of the footprint, it's generally the length of the read. Some of our reads are protected all the way to one end, but not the other, and those have a longer protamine profile, but more spread out. And then about 2/3 of our reads actually can give us the length of the protamine block from end to end, which is quite a lot. And we see a nice spread, a pretty wide variety in the sort of length of the protamine blocks in general, and including in sperm a peak where we expect to see mononucleosomes, which we didn't see in vitro, again, because we don't have nucleosomes there, which is nice. The lacunae also vary quite a lot in size. So this is a histogram of the distance between protamine footprints. So this is the lacunae I was talking about. Most of them are pretty short, on the order of about like 300 to 500 base pairs, but some of them can be quite long, up to kind of multiple kb. So because these lacunae are long, and because the way we define some of them are long, because the way we define them doesn't have anything to do with whether or not there's a nucleosome call in there, we can now ask, okay, in these gaps between the protamines, do we see nucleosomes? Like, are there nucleosomes there in the way that people have been talking about for a while, and what might they be doing? And so this is a plot of the lacunae binned by size, so shorter over here and longer over here. Um, and then the number of nucleosome footprints within those lacunae. And the main point here is that even as you get up into pretty high, pretty like large accessible regions, there are very few nucleosomes. It's not like they're retaining arrays of nucleosomes. They're retaining like maybe one or two, regardless of how big the lacuna is. And so this is kind of what we think is happening, is that the protamines come in, something's stopping them and kind of holding open that region. There might or might not be a nucleosome in there, but it doesn't matter that much overall for actually setting these things up. We could then look at how they are set up. So we also collected data from whole testis, which contains the different sort of stages of spermatogenesis. And what we see there, I'll skip to the slides. I think it's a little bit clearer. What we see there is that we actually can see blocks of protamines being set up pretty early. And this is we binned our reads based on protamine content as sort of a pseudotime, with the assumption that these cells are gaining protamines as they go through development. And there's a somatic tissue here for comparison. And as they accumulate protamines, they also lose nucleosomes, but they actually retain a relatively high nucleosome density until pretty late. So it implies that actually the protamines are coming in first. You keep the nucleosomes there, and then something stops the protamines, and then you lose the nucleosomes. And that sometimes leaves accessible DNA and sometimes doesn't. Okay, so what everyone wants to know is, where are these accessible regions? Where are the lacunae? They are more frequently at TSSs than we would expect by chance, but they are highly variable. So what I'm showing here is among TSSs, among promoters, the 5% of a sampling of reads from the 5% most accessible TSSs and the 5% least accessible TSSs. You can see that even in the most accessible ones, there's still about half the reads that are just fully protaminated. So there's no region of the genome that's always accessible, as far as we can tell, in every sperm. There's just a huge amount of heterogeneity from sperm to sperm. However, I'm showing like purposely suggestive examples here. This is a case of an accessible TSS, SOX2, and the inaccessible TSS, which is an olfactory receptor. There is some bias in the types of genes that tend to be the types of TSSs that tend to be accessible versus not. The biggest bias has to do with expression in the testis. So genes that were transcriptionally active during spermatogenesis leading up to the time of protamination tend to lack protamines at their TSS. There is also some bias towards specific histone modifications, even accounting for the fact that in order to have a histone modification there, you have to have a nucleosome there, so it must be a lacuna. But there's differences between the types of histone modifications that are more likely to be there. And we see K27 acetyl frequently, sort of corresponding to the transcriptional effect, as well as bivalent nucleosomes, which is something that was pointed out using ChIP-seq data again 15 years ago or so by Suhamud. So this is nicely at least confirmatory of that finding. And then the regions that do have lacunae, that do tend to be accessible, tend to be enriched for developmental functions, while those that are not accessible are these kind of multi-copy structural gene families. So we think that what's going on is regions that are transcriptionally active, that have particular chromatin states during those late stages of spermatogenesis, basically hold back the protamines as they're coming in. The protamine coat stops getting laid down, and then the transcription stops, and you lose the nucleosomes, you lose the transcriptional machinery, and you're left with this like lacunar space in there. Along a similar theme, we see something similar with transcription factors. And specifically with CTCF, actually, this is the case that we've looked at the best. So when we looked at sites that are bound by CTCF in spermatogenesis using ChIP-seq data, we see a nice strong CTCF peak. This is protected by, or this has a subnucleosomal footprint in our Fiber-seq data, high nucleosomes. And this is, again, testis reads binned by protamine content. So like this kind of pseudo time across development. We see the CTCF protection, that footprint there all the way through spermatogenesis until the very end. And this protamine kind of dip that shows up and stays there even as you get to very high levels of protamines. And then if you zoom in the sperm, the CTCF is gone. The CTCF left. But it left this scar in the protamine coat. So again, there's this gap in the protamines. It's a memory of what was there. It's not an indication that the CTCF itself is there, but it's a memory of what was there leading up to the mature sperm. The last thing I want to say, and I'm conscious of the time, is we also were lucky to get sperm from the HG002 donor, who's the donor who underlies the telomere to telomere reference sequence, which means that we could actually look at centromeres and telomeres as well. And what's shown here is different repeats and their association with telomeres, pericentromeres in the core centromeric region, and then nucleosomal footprints, short protamine footprints, and long protamine footprints. The punchline here is that telomeres have a lot of are well protaminated, but centromeres retain a lot more nucleosomes. This is consistent with previous observations that SEMP-A is retained in sperm, and we can actually see a SEMP-A footprint in the sperm. SEMP-A footprint is a little bit shorter than a standard nucleosome footprint, and we can see it there. The cool thing is that this is in the centromere dip region, which is a hypomethylated region where the kinetic core attaches. This is generally defined based on DNA methylation, 5-MC methylation. And we found that, yes, this region is hypomethylated in sperm, but the hypomethylation zone in sperm extends really far beyond the centromere dip region, this kinetic core attachment region. If we zoom out, this is really dramatic. So in somatic cells, it's really only in the CDR. In sperm, it's multiple megabases around the CDR. So this is not what's defining, DNA hypomethylation is not what's defining the CDR in sperm. We think it might actually be the SEMP-A retention that we're seeing there. So this kind of epigenetic definition of the centromere. So basically what I told you is that in sperm, the paternal epigenetic information is kind of flattened and simplified into these lacunae, where everything kind of gets boiled down into a gap in the protamine coat. Some of them contain nucleosomes, some don't, and they're really variable from sperm to sperm, but not random. There are patterns in where they are and where they aren't. And then the very last thing I wanted to show is, does this matter? We don't know the answer yet. We have a little bit of an indication because we looked at a few extra samples that were either normally fertile or infertile because they were very low motility. And we see surprisingly a really strong anti-correlation between protamine content and motility. So these they're all nicely protaminated, but and they have 86 to 96. Still lots of protamines, but the less motile sperm are actually the ones that have higher protamine content. And we see an inverse pattern when we look at nucleosome occupancy. So it seems that actually there's this like a Goldilocks zone where you want a certain amount of protamines and a certain amount of nucleosomes, but you don't actually want to go too far outside of that. Or it's going to result in infertility. And there's this little bit of an exception to the rule, which I don't have time to talk about, but would be happy to follow up with if anyone's interested in. So this is the same thing again. We see lacunae in the protamine coat. They're variable, but not random. They're associated with transcriptional activity and specific histone modifications. And it seems like there's an optimal balance of protamines and lacunae that's important for sperm function. Okay, and then lastly, I'll just thank the people who did it. So again, in my lab, this was led by Rachel Hewers, an amazing grad student, and the outstanding collaboration with Andrew and Tommy, and then a variety of people at University of Washington, and Chris and Adam, who helped us get the HG002 sperm. The sperm was the sperm samples came from the Yale Fertility Center with a great collaboration with Emre, Ahmet, and Jihan. So thank you for listening, and I'll happy to answer questions.
Thanks, Bluma, for the great talk. There is one question in Q&A, which is all somebody asked. I was wondering about your effect size that sometimes seem quite small but might still have high impact in sperm, as I'm not familiar with chromatin in sperm, but I'm still curious about these effects. I was wondering how these sometimes small observations affect the cell.
Sorry, actually, could you repeat it? I just was trying to get
It's in the Q&A box. So,
yeah,
yeah.
okay, yeah.
It's fine.
Um,
They're wondering about the effect size that sometimes it seems quite small, but may still have high impact in a sperm. Because I think they're out of this field about chromatin in sperm, so they were curious about if these small observations affect the cells in an impactful way.
I guess so the effect size meaning like the amount of difference in the protamine
I
content
think
overall.
so. Yeah.
Yeah, no, that's a great question. So we first of all, we don't know in many of these cases what the actual functional effect is. If you do or don't have like this level, a particular level of protamines at a particular type of region.
Mhm.
For that last slide I showed where we see a correlation to motility and immotility. Um, yeah, I mean, I think like the this is globally, so even though it's a small percentage overall, it's going to affect actually quite a lot in terms of absolute numbers of protamines and nucleosomes. And my best guess right now is that this is something physical rather than something about specific gene regulation, that you need just a certain level of compaction or a
Mhm.
certain level of openness spread throughout the genome at some number of regions. And if you go outside that threshold, it's going to be hard to reprogram at fertilization. But this is stuff that we have to test.
Yeah.
Yeah.
Ben also had a question. Ben, do you want to unmute yourself?
Yeah. Hey, that was a really cool talk. Good to see you again. I was wondering, so with the CDCF and these other sites that are accessible in the sperm, is it possible to try and look and see post-fertilization? Like, do you think these are getting bound by factors before the deprotamination that takes a few hours?
Yeah, I would love to be able to answer that. So that's on the list of things to look at. That would be cool. That would be a cool like implication. We can't test it in humans.
Yeah.
So the next the next step is to go into mice, I think, and start to look at that. But and it's very quick. First of all, it's very variable and very quick. So like it would have to be something about the dynamics, where if you slow down that nucleosome like entry even a little bit, that throws off the whole program because all of this is happening very fast. And then the question is, does it matter? Are there specific places where it matters, or is it just that you need some number of them, which would also be interesting to test.
Yeah.
Very
Uh,
cool.
there's one raised hand, David Miller. Jackson, can you unmute David so they can ask? Yeah, David, you can ask your question now if you unmute yourself.
Can you hear me?
Yes.
Great. Okay. That was a great talk. Thank you very much for letting me listen into it. I'm wondering whether these differences between lacunae and protamine-bound regions have something to do with the work we've been doing and others have done, looking at differential chromatin packaging in sperm, isolating chromatin with different salt concentrations, and then coming out and doing the sequencing on them. We do see evidence of differential packaging with developmental genes coming out in the low salt regions versus less developmental stuff, more structural stuff in the higher salt. Sounds to me as though we're looking at something similar but using a different approach. I'd like to get your comments on that. And also greetings from Poznan in Poland, by the way, when I'm speaking to you from.
Um, uh, Sorry, I didn't hear the last comment, but yeah, no, absolutely. I think that there's people been working on this, as you know, for a while. This is not like a new thing. And everyone tends to converge on this kind of, there's developmental genes that are differently packaged than the rest of the genome in sperm. And I think our data supports that. I suspect it's consistent with what you guys have seen. What the finding here is that the surprising finding to us was that how much variability there is from sperm to sperm. So it seems like maybe you need some set of developmental genes to be differentially packaged, but it doesn't always need to be the same 20 or 50 or 100 genes. It can be some subset of those. Once you're at a certain threshold, then you're okay, I guess. But yeah, no, I think it's totally consistent.
I think there might be some kind of a common framework,
Yes.
but then there's variation within that.
Yeah. And the data that we don't have, by the way, is, we see these variability between sperm. We have no idea which of these sperm are competent for fertilization. So it may be that there's only a subset of them that could actually generate an embryo, but we can't tell from our sequencing data which those are.
Yep.
Thank you.
Thank you, David. So we have a glitch and all our Q&A questions are gone, but our time is almost to the end too. And anybody, like there were like 4 or 5 questions in Q&A. Those people are welcome to email Bluma. I think
Yes,
she will
please
be
email
more
me. I'm totally happy to.
answer them. And I'm so sorry for the glitch, everyone. Yeah. And so that was our seminar for today. And thanks everyone for joining and hope to see you all in 2 weeks. And thanks Bluma and Sidhu for their great talks.
Thank you.
Thank you. Bye-bye.
Thank you.
Bye.
Bye, everyone.